So thank you very much. Good morning again, and I will do my best to introduce something about not only pleurothallid orchids, but also about evolution of some genome-wide traits. So firstly, I will have to explain what are these genome-wide traits, especially those which we are interested in. And the most interesting for us is probably endoreplication. This is something what happens in many plants. It's common in plants that cells which plan to differentiate, they replicate their DNA as shown here. Sorry, no, I don't like that. Which replicate DNA as shown here, but without cell division. So cells which are going to be some somatic cells to differentiate, they are increasing their individual ploidy levels. So then you can find some cells with, for example, this amount of DNA in leaves. And this is really common in plants, but only in orchids. It's possible that some species can choose only some species-specific selected part of the genome, which will be replicated in this case of cell differentiation. Naturally, if the cell plans to divide, it will replicate whole genome. But if it plans to remain some somatic cell and to endoreplicate, it selects only part of its genome to be replicated. This is really something unique, which we know only from orchids. Not all orchids do that, but some of them are doing. It's called partial endoreplication to distinguish it from this conventional endoreplication. And this is something what we would like to understand a little bit more. And we are also interested into other genomic-wide traits or genome-wide traits. On the left, it's genome size. On the right, it's proportion of GC base pairs in genomes. And this is across orchid phylogeny. Our first insight into this, where we mapped this on some phylogeny, and you can see some patterns. There is some variation. And maybe for this introduction, it can be interesting to highlight that orchids are one of the most variable plant family in respect to genome size. And they are absolutely most variable in GC base pairs content. They are pushing limits for whole angiosperms on both sides. They are exceeding these limits. So it's really something extraordinary. And we would like to study this a little bit more in one smaller group than orchids as a whole to be able to do some more deep analysis. And we choose Pleurothallidinae, which are really nice, and they have some features which are interesting for us, especially they are variable in our genomic characters which we are interested in. There are species with conventional endoreplication, and together with many species with partial endoreplication, they are somehow mixed in between in phylogeny. There is some variation in genome size in base pairs composition. So this seems to be really nice group to model this. But it has also some disadvantages or some challenges because it's really a huge group. At this time, it has more than 5,000 described species, and it's expected that it should have around 10,000 in real. And it's only 20 million years old. So if you compare these two numbers, you can say that this is probably the largest recent radiation in angiosperms. So it's really something challenging at this level. And well, our first insight into this was phylogeny based on HybSeq approach. And our first, which we published in 21, was with 337 species across the subtribe Pleurothallidinae. And this is because to understand evolution and to understand behavior of other genomic traits, we need some phylogeny to be able to work with that. So this is prerequisite to study it. And we need really good phylogeny, so we decided to design probes specific to Pleurothallidinae. They work somehow nicely if we are working with just one clade, but at this level of the whole subtribe, together with some outgroups which are not here, we were able to align just 168 nuclear and 288 plastid loci. But still, this was enough to distinguish some main clades and some nice relationships. But still, there are some parts which are not nicely solved, but it's something. And I can show some examples from this phylogeny in a little bit more detail. This is Pupulinia. It was proposed. You are smiling. I don't know why. It's a really nice plant. I can understand. It's this one. And this one was proposed to be separated from this one in 2020 based on morphology to trade this as monospecific genus. And we found this is true. It really has its own lineage here somewhere in the base of the subtribe. Yeah, it's funny plant. And what is extremely funny is that this single lineage with just one species, which we know now, it's more than 15 million years old. If you compare it with the age of that whole subtribe, 20 million years old, and the diversity of more than 5,000 species, this is, I think, really interesting to see that this is disproportional. There are some lineages which are not diverging and others which are diverging a lot. It's similar to orchids as a whole. So another example could be Speclinia, because based on morphology, these three plants are recently proposed to be single genus Speclinia. And we found even in our first phylogeny that they were separated into 3 clades. So we decided to improve our sampling and improve our analysis to understand relationships within this clade, which contains Speclinia. And well, we did some work. This we are now finishing, so it will be submitted really very soon. So hope you will be able to see it published also soon. We will see. And this is focused just on one clade. We call this clade D, and this is the clade which contains this genus Speclinia. And we found that really Speclinias form 3 clades. They are marked with these red dots. So one is something which is in fact Scaphocephalum, another one is Sarcinula, and then is the rest of Speclinias forming its own clade. And if you know these plants, it's also clear that Andreatea is nested within Muscarella. This was recently proposed with ITS. So we can see it here that it's really true and it makes sense. This is from Astral, and in this case, where we are working just with one clade, this is based on 630 nuclear genes. So it has a little bit better data set. And we did a lot of analysis, which I will skip for now. I can just show these SVD quartets result, and you can see that the main clades are quite nicely separated. It seems to be resolved. There is some issue in Sarcinula, which is somehow connected with Speclinia, maybe some ancient hybridization or something. What could also explain that if you use just 1 or 2 loci to see some phylogenetic relationships, you can be just, let's say, lucky, and you will got everything clustered together into one clade. But this may not be true if you see the prevailing topology of the trees. So this is how it looks, and this is more or less resolved, and it works nicely. Another situation is in Dracula, because we also decided to improve a lot our sampling in Draculas, because they are really interesting for understanding of evolution of the genomic characters, which we are interested in. So we decided, let's make phylogeny of Draculas to be able to study behavior of these traits more. This is what we got with Astral. You can see a lot of red color in these pie charts, so it's not very nice. Even that we were able to do this for more than 700 genes. So this is not really nice. We did, once again, I will skip some analysis which we did, but just very briefly, if you look on gene trees, which we have more than 700 of them. These are just 2 randomly selected. They have really bad supports, poor supports. And it seems if you look on a lot of them, that there are just some random pairs or small groups of species which are clustering together in one gene with some nice support, but in another gene, another groups are clustering together, probably just randomly selected from that mess of no sense what is there with poor supports. So if you use this, for sure it's only what you have with these data. So if you use this to calculate species tree, it's like that. This is not just astral species tree. This is from SVD quartets. Once again, so you see that there is really mess, and our data are not able to resolve these relationships. So just to understand this, this here it's outgroup. So it's also strange to see it here. But well, this is not working with our probes. So now we are generating another set of sequences in hope that that set of sequences will be able to resolve it, but we are still waiting for results, so I cannot show you more with Draculas. But it just seems that these 750 genes is not enough to resolve this phylogeny. They probably radiated very, very quickly in a short time. So let's go back to these genomic characters. This is just to go back to that. This is mapping of genome size just for pleurothallids. This is on our original data set of 300 species. At this time, we are still improving the phylogeny. So at this time, we have nearly 800 species in one astral tree, which is crazy. But well, this is for that 300 species. So there is some variation in genome size. But what is more interesting, it could be some relationship of the genome size with that partial endoreplication. So this is for that 300 pleurothallids. On the vertical axis, it's genome size. And the first box plot is for all taxa together. So there is some variation, why not? Second box plot is only for species with conventional endoreplication. So you can see they are somewhat smaller, and they have not so large variation in genome size. Also remember that this is on logarithmic scale. This is for species with partial endoreplication. So those species with partial endoreplication have larger genomes and higher variability in genome size. And this is maybe at the first look a little bit hard to understand, but it's not so crazy. This color, which I am probably not able to name even in my mother language, it's somewhat related to orange. So I will call it orange for now. Sorry for that. I don't know how to say it even in Czech, so well, it's orange for that. And so with this orange color, it's highlighted only that part of genome which is endoreplicated from these species with partial endoreplication. With this gray color, it's only that part of genome which is non-endoreplicated from the same species. So it's virtual separation of the genome into two parts, which already makes sense. And you can see that this part, which is non-endoreplicated, it's conserved in evolution, and it has similar size as the whole genome of that species with conventional endoreplication. But that part, which is not endoreplicated, this is what is highly variable, and what is probably very quickly evolving in evolution, and what is creating this higher variability in species with partial endoreplication. These are a little bit older pictures done for orchids as a whole from our 19 paper. And the first picture shows something very similar or nearly the same. It works also for orchids as a whole. Species with conventional endoreplication have smaller genomes. Those with partial endoreplication have larger genomes. But here is also some comparison of GC base pairs content in genome. And you can see that it's conserved in species with conventional endoreplication, but highly variable in species with partial endoreplication. This is the same in different projections. This is divergence from median, so you can see that the divergence is higher in species with partial endoreplication. That's not surprising. So this variability in GC base pairs content is created by those species with partial endoreplication. So try to find some other relationships to understand what is there. We tried to correlate genome size with chromosome counts. It's nearly nothing there. Maybe something very weak for species with conventional endoreplication, but mostly nothing. Here it's correlation with repeatome content, and there is really nice correlation. It shows that what creates differences in genome size in this younger group is mostly accumulation of repetitive elements in genomes. And well, this is a little bit crazy picture, hard to understand, but very informative. So on the vertical axis, there is size of repetitive elements in genome, how large portion of genome is occupied with repetitive elements. On the horizontal axis, there is size of genome. And in the case of black dots, this is for the whole genome, just for species with partial endoreplication for now, but it's whole genome in the black. And there is some relationship. Well, why not? In this orange, no name color, there is also some relationship, but weaker. And for that, it's that part of genome which is endoreplicated. For that part which is not endoreplicated, there is much nicer relationship. Really, really nice. And this shows that genome expansion in these plants is probably done by accumulation of repetitive elements in that non-endoreplicated part of the genome. And we can see very similar thing on a little bit different projection in these graphs. Here on horizontal axis, it's genome size. And let's start with this chart. Here it's on the vertical axis is fraction of genome. It's the size of that fraction of genome which is projected there. It means in this somewhat orange color, endoreplicated part, in this gray color, non-endoreplicated part. And you can see there is some relationship for that endoreplicated part, but just weak. But what correlates really nicely with genome size is that non-endoreplicated part. So this is what creates the variability. And these three other charts are for three separate genera, Dracula, Masdevallia, and Lepanthes. When at smaller phylogenetic scale, you can see that endoreplicated part is more or less conserved, and what is proliferating there is the non-endoreplicated part. So to conclude, orchids are really hyper-diverse in genomes respective to genome size and especially GC base pairs content. This is really outstanding. This variability is caused by those species with partial endoreplication, not by that with conventional endoreplication. This is probably because that non-endoreplicated part of genome accumulates repetitive elements, and they can shift that GC base pairs composition to some crazy values, and that endoreplicated part is conserved. So this is, and thank you for your attention.
Thank you for your talk. I don't understand. Could you briefly explain how do you measure genomic size or nuclear genome size of the non-endoreplicated versus the replicated portion?
Yeah, we use flow cytometry to measure this. And you can, well, if you use, for example, a leaf of a plant, you will have population of cells with different ploidy levels. So you will get a histogram with several peaks. The first one, if you choose good leaf, and you will be lucky, the first peak will be 1C nuclei. Then you will get peak with nuclei after first round of endoreplication. In species with partial endoreplication, it's not exactly 2C, but sorry, the first peak is 2C, then it's 4C. Sorry, but this is not exactly the double. And if you see these peaks, they are somehow shifted. They are the first peak is somewhere, the second one is somewhere, and the distance between peaks is not the double in species with partial endoreplication. And from this, you are able to calculate how large portion of genome is endoreplicated from distances between these peaks, if you understand. And this is how you can recover this. You can also do it for separate staining with DAPI and propidium iodide. So you can also estimate proportion of GC base pairs separately for these parts of genome, if you are familiar with this.
Jan, thanks. I have a comment and a question. And the comment is, well, I'm glad Pupulinia is supported.
Yeah.
Of course.
It's published in 21.
We did not only base it on morphology. We did have DNA
Yeah.
data. We didn't publish it when we published it, but
Yeah.
we knew about it. But my comment was more, my question was more related to what do you think is driving diversification in pleurothalids and those differences that you're seeing. So why do we have an old clade with one species and even younger clade that has 1,000? What is driving those differences? What do you think is causing that?
This is, yeah, thank you, Adam, for your question. It's a really nice question. And I would like to also know the answer, but I don't. We also really, we don't know. This is crazy, and you can see the same pattern in orchids as a whole. There are some lineages which are now represented even with single species after some tens of million years of evolution. For example, at the base of Epidendroideae, and there are other lineages which diverged similarly to pleurothalids. So it's crazy. And there is something known in publications, some correlation with evolution of some characters, but I think we are still far from understanding. And I can just tell that we already tried to model diversification rates in pleurothalids just on this subset of 300 species. It's too small subset. But we tried and to correlate it with our genomic features. And we found that probably it seems that species with partial endoreplication are diverging faster than those with conventional endoreplication, or that maybe that places of tree where this is switching between are somehow speeding the diversification. But this needs better sampling. At this time, we have nearly 800 species, and we want to make some diversification analysis when we will have at least 1,000 species of pleurothalids to make it meaningful. So, yeah, I will not tell more about that.
Okay, we have time for one more question.
Actually, I think you just answered it. I was going to ask whether there are any ecological or morphological correlates to, genome size.
Yeah, of genome size, they are. There are some. Well, some of them are already known. Some of them are somehow vaguely supported. But I would highlight maybe just one. Species with larger genomes are more frequent in colder areas, for example, more far from equator or in higher mountains in tropics, while species with smaller genomes are more frequent in warmer regions. So it means lowlands in tropics. It may not be a lowland like 100 meters above sea level, but maybe 1,000 above sea level. It's still a relatively lowland here if you compare it with high mountains. So, for example, this is some differentiation. It seems to also work if you have some dormancy period caused by climate, for example, some periodically dry areas. There are usually species with a little bit larger genomes than in areas with constantly moist conditions which can support growth for the whole year. So there is something. We still need more analysis to understand it better. We are also working on that.
Thank you so much. And let me please join me in thanking Jan again.